now let's look at the second part of this first subtopic, and this will be about the monohybrid cross. The first type of cross that Mendel usually did was this monohybrid cross. Okay, what is the meaning of this? In this particular cross, Mendel observed only one particular characteristic. So as usual, in Mendel's experiment, he started with two parent individuals that are true breeding parents. True breeding parents will be referring to individuals that are homozygous. Okay, that are homozygous. So Mendel chose this dominant homozygous individual, a tall plant, in which we can see the genotype is capital T, and another parent individual will be this homozygous recessive individual, small t. Okay, and the phenotype will be short plant or dwarf. So this will be the parent generation, and from these two parent individuals, this will be the types of gametes that can be produced. Okay, from this tall plant, from this tall parent, this type of gamete can be produced, which contains the capital T allele. Meanwhile, from this homozygous recessive individual parent, this type of gamete can be produced, which contains this small letter T allele. So if we combine these two types of alleles, then the F1 generation will be produced, and all of the F1 generation will be having this same type of genotype, heterozygous, capital T, small t, and the phenotype will be tall. All of the offspring, 100% of the offspring will be tall plants. But Mendel did not stop at this level. Mendel continued with the self-cross, in which Mendel took one individual from this F1 generation, and Mendel crossed with another individual from the same generation. So F1 crossed with another F1 individual, and this will be the gametes. This will be the gametes from this heterozygous individual, in which one type of the gamete will be containing this capital T allele, the dominant allele, and another one will be having this small t allele, the recessive allele. The same goes for this other individual. Okay, so if we cross all these gametes together, then we will get this F2 generation. This will be the genotypes of the F2 generation individuals, in which we can see that 1 out of 4 will be having this homozygous dominant genotype, capital T. Meanwhile, 2 out of 4 will be showing this heterozygous genotype, capital T, small t, and 1 out of 4 will be showing this homozygous recessive genotype, small t. So if we do a ratio for the genotype, we call it as the F2 genotypic ratio, it will be 1 to 1. Okay, 1:2:1. And if we look at the phenotype for this F2 generation, okay, we will be able to see that 3 out of 4, 1, 2, 3 out of 4 will be producing tall plants, will be having tall type of phenotype. Only 1 out of 4, about 25%, will be showing this short characteristic. So if we do a ratio known as the F2 phenotypic ratio, we will get this type of ratio, 3:1. Another example will be about the fruit flies. So Mendel only performed his experiments on the garden pea plants, but later on scientists performed this monohybrid cross on the fruit flies, Drosophila melanogaster, and so the same type of result still can be obtained. So for example, if we are looking at these fruit flies, Drosophila, we can observe the characteristic of the wings. There are two types of wings. One will be the normal wings, another one will be the vestigial wings. Vestigial wings will be short and crumpled. So if we cross two individuals, okay, one having normal wings, this is actually a dominant type of phenotype, Okay, and another individual parent will be having the vestigial wings. This will be the recessive type of phenotype, the recessive phenotype. And so this will be the genotype of this individual, capital V, capital V. Meanwhile, for this individual, it will be the homozygous recessive, small v, small v. And this will be the gametes from these two parent individuals, okay, in which from this individual, one type of gamete can be produced containing this capital V allele, the dominant allele. Meanwhile, from this recessive individual parent, there will be this type of gamete containing this recessive allele of small v. So the combination of these two gametes will be producing this F1 generation, all having normal wings, but the genotype will be heterozygous, capital V, small v. And if we continue with a self-cross between this F1 generation individuals, okay, this will be the type of gametes, okay, in which from this individual of heterozygous condition, we will get these two types of gametes. One will be containing the dominant allele capital V, another type of gamete will be containing the recessive allele small v. Okay, the same goes for this individual over here. So if we cross all these gametes together, we will get the F2 generation. And we can see that 3 out of 4, 1, 2, 3 out of 4 will be showing normal wings type of phenotype. Okay, meanwhile, only 1 out of 4 will be showing this type of recessive phenotype, vestigial wings. So if we do this in the form of a ratio, so the ratio will be 3:1. Nisbah 3:1. Okay, so the phenotypic ratio for the F2 generation will be 3:1. And if we do a test cross, let's say we do a test cross on a heterozygous individual. This heterozygous individual is dominant because it is having a normal wings. It is having normal wings. But maybe we are not sure about the genotype, so we just cross this individual with a recessive fly having vestigial wings. So in this test cross, we'll still be getting the same type of result proposed by Mendel, in which the ratio of the offspring will be 1:1. And from this monohybrid cross, Mendel proposed the law of segregation. This is the first law of Mendel, in which the law of segregation tells about for one characteristic, it will be controlled by a pair of alleles, okay, because we know that one allele come from the father, another allele come from the mother. So one individual will be having a pair of alleles for one particular characteristic. Okay. However, these two alleles will separate during gamete formation and end up later in different gametes. So the law of segregation is actually referring to the separation of the two alleles, which initially are together. Initially, we can see that two alleles are together side by side due to the process of synapsis, maybe. But during meiosis, okay, due to anaphase 1 and later anaphase 2, we can see that gametes will be produced finally, and each gamete will be containing this type of alleles, in which the alleles just now are now separated into different alleles. So we can see that some of the alleles will be the dominant allele, but some other gamete will be having this recessive allele. So this capital A and small a alleles that are together at the beginning, now they are separated into different gametes. So that will be the first law of Mendel, known as the law of segregation. Okay, and so this is the summary for the monohybrid cross that you need to take note. Okay, so again, in a Mendelian monohybrid cross, okay, in a Mendelian monohybrid cross, normally the parents will be pure breeding parents, which are homozygous individuals. Example, we cross a plant with purple flower, okay, that are homozygous dominant, and another plant which will be having white flower, which is showing the homozygous recessive condition. Okay, so capital P cross with the small p. Okay, and all of the F1 generation will be heterozygous. So for example, the F1 generation will be having purple flowers, all of them, and the genotype will be capital P, small p, like this, heterozygous. And if we do a self-cross, between the F1 individuals, finally for the F2 generation, okay, we will be getting this type of phenotypic ratio 3:1. Okay, nisbah 3:1. For example, 3 out of 4 will be having purple flowers, and only 1 out of 4 of the F3 generation will be having white flowers. So the ratio will be 3:1. Meanwhile, if we do a test cross on a heterozygote, okay, a test cross on a heterozygous individual, so we will be getting this type of phenotypic ratio 1:1. Okay, in which the offspring from this test cross will be 1 purple and 1 white flowers. Okay, so please remember these two types of phenotypic ratios. And one thing that is also important about this monohybrid cross will be about the first law of Mendel, known as the Law of Segregation, in which we say that two alleles that are initially together, okay, will later on separate during gamete formation. Okay, so this is all about the monohybrid cross. Okay, next we will continue with the dihybrid cross.